Bueno, pues buenos días, bienvenidos a nuestro seminario institucional. El día de hoy nos acompaña el doctor Iván Martínez. Él es profesor asociado en el Instituto de Cáncer de la Universidad de West Virginia y el Departamento de Microbiología, Inmunología y Biología Celular de la Universidad de West Virginia. Él nació en la Ciudad de México, se graduó con honores en la Universidad Nacional Autónoma de México como biólogo, somos compañeros de generación. Él hizo su doctorado en genética molecular y bioquímica en la Universidad de Pittsburgh y en el laboratorio del doctor Salim Khan, estudiando las interacciones entre la infección del virus de papiloma humano y los microRNAs. Posteriormente, el doctor Martínez llevó a cabo estancias postdoctorales en el Departamento de Genética de la Universidad de Yale, en la Escuela de Medicina, en el laboratorio de Daniel Di Maio, en colaboración con el doctor Joan Steitz, estudiando la importancia de los microRNAs en el arresto del crecimiento celular. Posterior a su entrenamiento postdoctoral, el doctor Martínez ingresó a la Universidad de West Virginia en el 2013 y en esta universidad el objetivo de su investigación es el entender la importancia de los diferentes tipos de RNAs no codificantes en tumores humanos, así como en los procesos de infección viral. Algunos de sus hallazgos han sido publicados en revistas de alto impacto, incluyendo PNAS, Journal of Cancer Research, y también ha estado presentando en diversas instituciones de muy alto nivel, como la Universidad de Harvard, la Universidad de Göttingen, en el Max Planck Institute, y también él está muy involucrado en la organización de distintos encuentros internacionales, por ejemplo, el Workshop on Long Non-Coding RNAs, y también el Keystone Symposia de Small Regulatory RNAs from Bench to Bedside en Santa Fe. También el doctor Martínez es un miembro del grupo de trabajo en modelos de COVID de la Organización Mundial de la Salud desde el 2021 y en esta ocasión precisamente nos va a presentar datos acerca de su investigación, acerca de la importancia de los RNAs no codificantes lineares y circulares para el cáncer humano. Bienvenido, Iván, y muchas gracias por acompañarnos.
Muchísimas gracias. Gracias. Gracias, Alfredo. ¿Se me escuchan? Sí. Perfecto. Bueno, antes que nada quiero agradecer a Alfredo y por supuesto a la IMEGEN por darme la oportunidad de presentarles un poquito de lo que estoy haciendo allá en West Virginia University. Desafortunadamente estoy más acostumbrado a dar la plática en inglés, entonces me perdonarán, pero lo voy a tener que dar en inglés. Si hay alguna pregunta, por supuesto estoy abierto a que me interrumpan en cualquier momento. Pero pues sí quiero enseñarles un poco de lo que estamos haciendo allí en el laboratorio. No les tengo, por supuesto, que recordar the central dogma of molecular biology, where we know that the DNA is when we keep all the genetic information, and then, of course, it's transcribed to RNA, and then from RNA, it's translated to proteins. So this is the dogma. We've been following the biomedical research for the last 60, 70 years. But something happened after the sequencing of the human genome in 2000, and not only the human genome was sequenced, but other organisms were sequenced. And when they're looking at the DNA and also the transcriptomic, they realized there was something different. Why? For example, if you imagine that blue dot there is this blue dot here is all the DNA that a bacteria has, yeah? How much of this DNA is actually transcribed to RNA? In bacteria, we're talking about close to 98% of their DNA is transcribed to RNA, okay? How much of that RNA is translated to protein? We're talking about 95% of the RNA in a bacteria is translated to protein. So very simple organisms, very efficient what they're doing. But when we talk about, for example, insects, how much of the DNA of an insect is transcribed to RNA? It's almost the same. It's talking about 90%, 95%. Between 90 and 95% of their genome is transcribed to RNA. How much of this is actually translated to protein? 70%. Only 70% of the RNA that they produce is translated to protein. But then, of course, comes humans. How much of the human genome is actually transcribed to RNA? And we're talking about still between 85 to 90% of our genome is transcribed to RNA. How much of this RNA is translated to protein? and that's the thing I love to show to everybody, because a lot of people don't know this. 1 to 2%. 1 to 2% of our RNA in our cells are translated to proteins. All our biomedical field is based on this 1 to 2%. So we technically have, and of course people thought that was just garbage that we have there, junk, DNA junk. But now we know that the non-coding RNA, so the RNA that doesn't translate to protein, is a dark matter biology. We know it's there, but we have absolutely no idea, most of them, what is their function. But we have it. So that's really my passion, to understand all these non-coding RNAs and what is exactly their importance in human diseases. This is a list of course, all the different types of non-coding RNAs. I'm not going to go through them. But mostly non-coding RNAs are divided in small non-coding RNAs and large non-coding RNAs. Yeah? The very first one that was discovered was microRNAs. Yeah? This was discovered by Dr. Victor Ambros. Interesting. He discovered in 1991 in C. elegans. Nobody cared about that time. In 1991, published a paper saying, oh, I discovered these small microRNAs in C. elegans, and these little worms. He's like, okay, that's fine. Who cares? And then 10 years after, was not until 2001 that they discovered in humans. Now we know that we have between 2,654 microRNAs in our human genome. Yeah? But today I'll be talking mostly about long non-coding RNAs. Long non-coding RNAs is a group close to 57,000 in our genome. And it was not until 2009 that Dr. John Green, at that time he was at the Broad Institute, published this paper showing that they discovered at least 1,000 human long non-coding RNAs. Yeah? And the other one I'm going to be talking about a little bit today is circular RNAs. Circular RNAs, I'm going to show you where they come from, but we have something in between 92,000 circular RNAs in our genome. And I want to put this just to show you. We're talking about tens of thousands of non-coding RNAs compared to the 20,000 protein-coding genes that we have in our genome. And how much of these non-coding RNAs we know their function, I will not say no more than 2 or 3% of them. So we have a universe to understand, to investigate, and see what is their function. And of course, now we know that these three groups, microRNAs, long non-coding RNAs or lincRNAs, and circular RNAs or circRNAs, they are part of the hallmarks of cancer. Any type of cancer, any hallmark of cancer you can think of, immortalization, proliferation, survival, apoptosis, avoiding apoptosis, anything like that, you can potentially find a specific non-coding RNA working in that pathway. But now we're going to be just talking about long non-coding RNAs. This is the first story I'm going to tell you. It's one of the stories that is mostly developed is the one that has several years working in my laboratory, and we have, I think, very interesting data. So, but what is a long non-coding RNA or lincRNAs? These are any RNA that is 200 bases long. So they can be 201 bases, so they could be 10 kb. That's, as long as they don't translate to a protein, is considered a long non-coding RNA. They don't normally have an open reading frame. They're mostly dysregulated in different diseases like cancer, and they have multiple functions. That's the interesting thing about long non-coding RNAs. The RNA itself has several different functions. For example, they can be RNA decoys. These long non-coding RNAs, they can bind to transcription factors and stop the ability of the transcription factor to bind to the DNA and start transcription of this gene. Yeah? They can work as microRNA sponges. They can interact with microRNAs, and now the microRNA cannot interact with the messenger RNA that was targeting. They can work as protein bridges. There are several studies showing that different proteins have different protein complexes. They work together, but it was not until recently that they realized these long non-coding RNAs work to bring them together. They can work as chromatin modifiers. There are these long non-coding RNAs, in the moment they're getting transcribed, they recruit proteins, and literally they change the structure of the DNA to turn off and turn on genes. And They can also work in translation and division, splicing modulation, and they can degrade messenger RNAs. Yeah? So again, this is just to show you that long non-coding RNAs have several functions. Just one long non-coding RNA could work in different ways. So one of the things I'm going to show you today, the project was mostly started by one of my PhD students, Tavia Brown-Miller. Now she's a postdoc at NIH. And right now, Emily Rice, my other PhD student, is trying to finish part of this project. Of course, I don't have to tell you that lung cancer is the deadliest of all cancers. It's the one that kills more people around the world. And we, of course, clinicians, they use different therapies to treat lung cancer. We're talking about surgery, chemotherapy, radiation, and most recently, immunotherapy. But something that people don't know too much is that, of course, in the moment that these lung cancer tumors, they develop resistant to radiation, there are different pathways that they are involved in these radiation-resistant pathways, that there are several genes involved in these pathways, but actually there are several non-coding RNAs regulating these genes. And there are several publications showing that. Some of these long non-coding RNAs, some microRNAs are important in producing radiation resistant in different type of cancers. But for us was which link RNA could be important in radiation resistant in lung cancer. So we started this project by screening a cell line called H460. It's a very common lung cancer cell line. And the thing we discovered actually was something interesting. One of the most highly expressed group of long non-coding RNAs after radiating this lung cancer cell line was a family of link RNAs called SPRY3. Yeah? So we said, okay, these are highly expressed after we radiate the cells. What happened after? Turns out this group of long non-coding RNAs are actually transcribed from the DYCY region of the Y chromosome, meaning these long non-coding RNAs are only male expressed because they come from the Y chromosome. So that was, for us, was actually very interesting to see, wow, this is, nobody ever really showed us before, sex-specific long non-coding RNA. We demonstrate that yes, if you radiate the cells, you leave the time, you actually take different time points and you see the expression of these 3 link RNAs increasing after radiation. But after doing this screening, we said, okay, this is just one cell line. Let's expand our findings to more male lung cancer cell lines. So that's what we did. We collect several lung cancer cell lines. They're all male. Some of them, the driver mutations were different, KRAS, EGFR. Some of them were actually recently, these are PDX cell lines that we obtained there at WVU, and we even include a normal immortalized epithelial lung cells. Yeah? One of the first things we did is like, okay, let's test which of these cell lines are already intrinsically radiation resistant or radiation sensitive. So we did that by colony formation assay, and we were actually able to divide in 2 groups which cell lines were radiation sensitive and which ones were radiation resistant. Yeah, so that was good. We have 2 groups of cell lines that we can, okay, radiation sensitive, radiation resistant. Let's look at the expression of these long non-coding RNAs. When we did that, something very, very shocking happened. You can see here, in the beginning, this is just the normal immortalized epithelial lung cells. The more radiation we give them, the more expression of these long non-coding RNAs. Yeah? Then all the radiation-sensitive cell lines, all of them, you can see these link RNAs, the more radiation you gave them, the more expression you have. Yeah? They were all radiation sensitive. What happened with the radiation resistant? No expression at all. 0. This thing that you see right there is just background. So when my students showed me this, I was like, I never saw something so black and white. so different. So we went back to the literature to see what could be happening in the radiation-resistant cell lines. So we went to the literature, and turns out, since the early 1990s, people knew that some male cancers can lose their Y chromosome. People always thought that losing the Y chromosome in different male cancers was just part of the process of carcinogenesis, there's just so much genetic instability that losing the Y chromosome, there's such a small chromosome, you lose it, nothing happened, nobody cares. So the first thing we did is we questioned maybe some of the radiation-resistant cell lines lost their Y chromosome. So the first thing we did fluorescent in situ hybridization, FISH, of DNA, using different colors probes. We have a blue light color for the centrosome of the X chromosome as an internal control, but we have two probes for the Y chromosome. We have a red dot for the P arm and the green dot for the DYCY region. That is exactly where these long non-coding RNAs are coming from. So when we did this FISH DNA, you can see this is the HBEC cell line. This is the immortalized normal epithelial cells, long cells, and you can see the Y chromosome right there, and the X chromosome right there, and the Y chromosome right here. Yeah? You should have that because it's a normal cell. It's just immortalized. When we did that in the radiation-sensitive cells, you can see, it's hard to see from here, but you can see there's several green dots showing that most of these cell lines, they don't only have one Y chromosome, they have several Y chromosomes. Yeah? When we look at the radiation-resistant, there was no Y chromosome. Gone. So we're like, wow, that's why there's no expression of these link RNAs. So we were very excited about that findings. But again, this is not telling me any functions. This is just telling me that the Y chromosome is there and these link RNAs are not present. So, and then of course we check female. These are female lung cancer cell lines that you can see that the expression of these link RNAs doesn't exist either. So of course we want to know if this link RNA family was actually involved in radiation sensitivity. Because again, this is just telling me that they're related, but that doesn't tell me that the function is real. So one of the first things we did was to knock down these link RNAs. We used shRNAs to knock down these link RNAs. And something interesting happened. When we designed an shRNA against SPRY3-2, not only we knocked down this guy, but we knocked down all the family. When we designed one for 3-3, that is this guy, we knocked down the entire family too. And this gave us some idea that probably this is not a family of long non-coding RNAs. This is probably a very long, one single piece of long non-coding RNA that for whatever reason is at that time, even now, is very hard to map the Y chromosome. So we believe that this is not a family, but this is a big long non-coding RNA. So we did it in two different cell lines. We were able to knock down the link RNAs. And then we did some cell viability assays. And I'm not going to go too much into the details, but the thing I can tell you is we did cell viability, we did apoptosis, and we can see that in the moment we knocked down these link RNAs, the cells that were more radiation, this is an H460 cell line, this is a radiation-sensitive cell line. In the moment we knocked down these link RNAs, the cell line actually became more radiation resistant. And the same for apoptosis. The moment we knock down these link RNAs, these cells have less apoptosis than when they have it. The next thing we did with an in vivo model, we introduced the cell lines into the flank of different mice, and then we measure by ultrasound the size of the tumors, and we actually were able to really identify every single one of these tumors and precisely look at the sizes of this in vivo model. And again, I'm not going to go too much in detail. We did, like the clinicians do, we did a specific treatment to these mice with different doses of radiation. And you can see here in the moment we knock down the link RNAs, the tumors actually grow more than the ones that were not treated. So that was giving us some idea that the presence of these linked RNAs give them sensitivity to radiation to the cells. But how to do the opposite? How to introduce these linked RNAs? Remember I told you, at that time the mapping of the Y chromosome was so hard that we were unable to clone every single linked RNA because we didn't have the entire sequence of these linked RNAs. So the only way we were actually at that time we were able to do it was introducing a bacterial artificial chromosome. I don't know, you guys know this, but the way they sequence the human genome was by cutting different pieces of the human genome and introducing them into these bacterial artificial chromosomes. So these are nothing else than plasmid that can keep big pieces of DNA. And we were actually able to identify a bacterial back that has the DYCY region where these linked RNAs are expressing from. That is actually this RP1188F4. So this region of the Y chromosome was in this back, and we know that that region was precisely where these linked RNAs are coming from. So the thing we did, we artificially introduced this back into A549 cell lines that they don't have the Y chromosome, these cells, as the ones that lost the Y chromosome. We introduced it artificially, and you can see here, we were actually able to recover the expression of the long non-coding RNAs. So we were really, really happy about this. Then the next thing we did the same thing I showed you before. We did cell viability. And in cell viability, you can see here that just by introducing this expression, these linked RNAs, in a radiation-resistant cell line, actually the cell became more sensitive to radiation, and also they became more apoptotic. So showing us the other side of the coin, that by introducing these linked RNAs in a radiation-resistant cell line, we make it more radiation sensitive. But what is the mechanism? What exactly these linked RNAs are doing? Yeah? So one of the first things we did was to identify the sequence of these linked RNAs, at least fragments of them. And something was interesting. We identified this very repetitive sequence that is the binding site for a specific RNA binding protein called IGF-2BP3. IGF-2BP3, or insulin-like growth factor 2 binding protein 3, is a RNA binding protein that really one of the main functions is an RNA stabilization. So these proteins bind to the messenger RNAs of oncogenes like CMYC and HMGA2 and give them more stability to the messenger RNA. So that way, these messenger RNAs can produce more protein. They've been found upregulated in different types of cancers, including lung cancers, and actually previously been implicated in radiation resistance. So we said, okay, this is going to be an interesting finding. So the first thing we did was to do immunoprecipitations using IGF-2BP3, and something that we were very happy was we were actually able to recover all the 3 linked RNAs by pulling down with IGF-2BP3, including the positive control HMGA2, but not GAPDH. Yeah? That shows us that IGF-2BP3 is actually binding to these long non-coding RNAs. So the next thing we did was to look at stability of the messenger RNA. You do that by introducing actinomycin D. You stop transcription, and then you look at the stability. So in this case, you can see here, this is the messenger RNA for HMGA2. In the moment we introduce artificially the linked RNAs, you see a drop of HMGA2 stability, meaning the presence of the linked RNAs is going to sponge IGF-2BP3 protein, and now the stability of HMGA2 is going to go down. But if we introduce an SI control, meaning if we knock down artificially the back, we were actually able to recover the stability of HMGA2, and we did that in different time points. Yeah? We did the same thing with CMYC. You can see here, CMYC drops after introducing artificially the links, but if we knock down the links, CMYC goes up, and the same for the rest. Yeah? that told us, and this didn't happen in GAPDH, that we know IGF to BP3 is not binding to. So these data demonstrate that these link RNAs are actually sponging IGF to BP3 and give them less stability to the oncogenic messenger RNAs. So the model that we had at that time, and that's the one we published in Cancer Research, was that cells, lung cancer cells, that lost their Y chromosome, they don't express these link RNAs anymore. And now IGF to BP3 is going to bind to the oncogenic messenger RNAs and give them more stability. There's going to be more expression of the genes, of the oncogenes. Meanwhile, in cell lines that they still have the Y chromosome, the low non-coding RNA is going to sponge part of them, and now you're going to see less expression of CMYC. So that's one of the first findings that we got from this low non-coding RNA family. But we're continuing because, like I told you before, these long non-coding RNAs not only have one function. They've been shown that some of them, they have 2, 3, 4, or even more functions inside the cell. So one of the things we looked was to do more RNA-seq. After we knocked down this link RNA, we identified several genes upregulated and downregulated by the absence of these link RNAs. We did some pathway analysis, and we identified certain genes, specifically genes involved in cell cycle regulation, involved in migration, involved in senescence, that were actually increased in their expression, or some of them they were actually decreased in their expression after losing the link RNA. One of the first things we did, we also did cell fractionation to know where these link RNAs are in the cell. And turns out these link RNAs are nuclear. You can see here. They are non-cytoplasmic. And there's a lot of long non-coding RNAs that they are nuclear. They can actually interact with their DNA. So one of the questions we have is, what if these long non-coding RNAs are actually directly interacting with chromatin, and that's how they can regulate the expression of these genes? So by using a tag-seq, we're actually able to identify which regions of the DNA is open or closed. We did that by knocking down the expression of the link RNAs, and we, of course, found some genes were increased, some genes that were decreased. But interestingly enough, some of the genes that I showed you before, like aromatase B, CDC6, CDC25A, look like the long non-coding RNA is helping to open that region. So actually, they're actually able to be transcribed. Not only that, we are right now in the process of moving into therapeutics, meaning we want to introduce artificially these link RNAs. We did it already in male cells. We already did it in female cells. A lot of times when I give this presentation, a lot of people ask me, what happened with female lung cancer cells? Well, we artificially introduced this Y chromosome male link RNA, and we actually were able to show radiation sensitivity. And we even introduced it in mice, in mouse cell lines. These are H1299s, these are human. But, sorry, I'm gonna go back to that. So this is just to show you that before I go and talk about therapeutics, we were actually able to clone each individual link RNAs. We were actually able to map it longer. We were able to clone it independently. And then we did some functional assays, meaning we introduced it artificially in cell lines like H1299 that doesn't have the Y chromosome. And by introducing these guys artificially, we actually saw a reduction in colony formation assay. We saw a decrease in cell migration. But here, interestingly enough, some of these link RNAs are more than the others, like 3-2, definitely gives less migration than the other two. And we even identify increase in senescence. By introducing these link RNAs, you can see here that in this H1299, without the link RNAs, we irradiate it, and it's hard to see it, but you start to see some green dots of cells that are senescence. But in the moment we introduce these link RNAs, you see these big green dots showing you that just the presence of the link RNA gives an essence to the cells. Not only that, we were actually able to identify the promoter region of these link RNAs, and we believe that they are regulated by two transcription factors, OC1 and c-Jun. C-Jun binds to the AP1 regions, and of course, we play with them, we knock it down, and we saw that the expression of the link RNAs after radiation was actually diminished, showing us that potentially these transcription factors are regulating the expression of the link RNA after radiation. There's evidence that these these transcription factors, OC1 and AP1, well, c-Jun, there are literature showing that their expression increase after radiation, so that makes sense for us. Not only that, we actually did this very interesting experiment where I show you most of our data is in a monolayer, growing the cells in a monolayer, radiating in a monolayer, and studying in a monolayer. But something really strange happened when we put these same cells inside the mouse. We saw a dramatic increase in the expression of the link RNAs. Then the thing we did was, okay, let's include spheroids. We include spheroids, and the spheroids actually increased more than the monolayer. And we even squeezed part of the monolayer, squeezed part of the tumor microenvironment of the in vivo tumors that were growing mice and put them in a monolayer, and we saw an increase of this, telling us that the three-dimensional structure and the tumor microenvironment in these cells are important in the regulation in the expression of these link RNAs. We don't know exactly why yet. So this is the other one I was just confused. This is the 3 LL cells. These are mouse lung cancer cell lines that we artificially introduced the link RNAs. And right now we are processing them, but I can show you right now that just by introducing them, we actually increase the amount of apoptosis in these cells, showing me that these very specific human link RNA, well, these link RNAs are present in primates, from gorillas, chimpanzees, and humans. We can put in a mouse cell, and now the mouse cells also become more radiation sensitive. So having all this data, that's why we're now moving to therapeutics. We're trying to artificially put these link RNAs in nanoparticles, the same nanoparticles that was used for the COVID-19 vaccine. The FDA approved. We're doing it right now in the lab. And we're going to inject PDX tumors that are grow next to on the side of the mice. We're going to inject it with these link RNAs, radiate them, and demonstrate that just introducing these link RNAs, we have a shrink of the tumor faster after radiation. But what is the clinical relevance, of course? So one of the clinical relevance, of course, we're trying to also follow is to look at the presence or absence of the Y chromosome. Like I showed you, you lose the Y chromosome, you lose the expression of the link RNA, you lose the expression of the link RNA, the cells become more radiation resistant. So one of the things we did was continue doing DNA fish, but we did DNA fish in tissue arrays. All these arrays that has 100 different patient tumors, and we look at the presence or absence of the Y chromosome. So we actually did that in several tissue arrays. You can see here our positive control was testicular embryonal carcinomas. You see the green dots, that's a positive cell that has the Y chromosome. Our negative control was cervical cell lines. So a cervical tumor that is, of course, a female tumor that doesn't have a Y chromosome. And we also did in male lung cancer tumors from patients and female lung cancer tumor patients. You see there's no in the females, but you see the green dots in the male, yeah? When we did that, something very interesting happened too. We have a software where the software can identify the green dot and give it a color to that cell. And if the cell has a Y chromosome, it's going to put it in a red color. But the cells that didn't have the Y chromosome is going to put it in a purple color. And you can see here, these are patient tumors, lung patient tumors, all male. And you see that some tumors, most of the tumors still have the Y chromosome. Some other tumors lost most of the Y chromosome. So that's intrinsically, we see that it's very, very different. And we are still continuing doing overall survival to show that the absence of the Y chromosome is you have poor overall survival. But what is next? After that, we want to use artificial intelligence. We want to use AI, because now there are a lot of machine learning classifiers that can help you identify differences in tumors, like how the tumor granularity is, the size of the cell, the size of the cytoplasm, the size of the nucleus. So our idea is there any way AI can help us to identify which cells lost the Y chromosome, which ones they still have the Y chromosome? That will be amazing, because now we can go with the clinicians and show them to the clinicians, this tumor from this patient, 90% of the tumor lost their Y chromosome, most likely their tumor is going to be radiation resistant. So we can give some advice to the clinicians how aggressive their radiation therapy should be for every patient. So in conclusions on this part of the linear RNA, linear long non-coding RNAs, I showed you that we discover a group of long non-coding RNAs that are highly expressed after radiation, but only the radiation sensitive, not the radiation resistance. I showed you that they don't express the link RNAs because they have a Y chromosome. We showed you that if we knock down the link RNAs, the cells become more radiation resistant. If we introduce the link RNAs, the cells become more radiation sensitive. And as I just mentioned, we're looking at tissue patients samples, and we're trying still to go deep into the molecular mechanism that these long non-coding RNAs are doing in lung cancer cells. Any question, guys, do you have before I jump into the circular RNAs? No? Yes. Sorry, that was first. Y me puedes preguntar en español, no hay problema. Perfecto.
Entonces, es súper interesante, estás respondiendo muchísimas preguntas que estás generando más.
Por supuesto, así es la ciencia.
Y en relación a este cargo no codificante, con la sobrevida de las pacientes, entonces ya las mujeres intrínsecamente tienen una peor sobrevida si estamos pensando que este está involucrado en la sobrevida de los pacientes.
Sí, esa es una muy buena pregunta y esa siempre me la han preguntado cada vez que doy esta plática. Porque, no te quito No. A lo que voy es, sí, o sea, uno pensaría que las mujeres, por no tener el cromosoma, este link RNA en el cromosoma Y, serían más propensas a tener problemas en cuanto a resistencia a radiación. Y resulta que es lo opuesto. Cualquier clínico te puede decir que si llega una mujer y un hombre con el mismo tipo de tumor de cáncer, del mismo tipo de tumor de pulmón, el simple hecho de ser hombre, tú ya tienes, tu overall survival es menor que el de la mujer. ¿Por qué? Porque aquí estamos hablando puramente de genética. Pero cuando ya estás hablando de overall survival, tienes que hablar de genética, de hormonas, de tipo de vida. O sea, hay tantos factores involucrados que lo más seguro es que todos esos en conjunto ayuden a las mujeres a tener el sistema inmunitario, por supuesto, es mucho más eficiente en mujeres que en hombres, y eso se ha demostrado. Las hormonas, el cambio hormonal también muchas veces ayuda a las mujeres en ciertos tipos de cánceres que a los hombres no tenemos esa facilidad. Pero al final de cuentas es una pregunta que no podemos, con este tipo de investigación no podemos contestar. Ahora, si las mujeres tienen estos link RNAs que podrían estar en el X chromosome, sí, y lo estamos buscando. Potencialmente a lo mejor ellas los tienen en el X chromosome y pueden compensar lo que los hombres tienen en el Y chromosome.
Otra pregunta es en relación al efecto biológico. Ya se demostró, tú también lo estás observando, que son muy importantes estos procesos asociados a cáncer en general. Pero, ¿tiene alguna participación en la replicación del cromosoma?
No que sepamos, no. Pero algo muy interesante, y esto no lo tengo en la presentación, claramente estos long non-coding RNAs y esa región de donde vienen son importantes no nada más en radiación, sino en otras cosas. Por ejemplo, tenemos preliminary data donde vemos que si utilizamos quimioterapia, estos link RNAs también suben de expresión. Pero no nada más eso. En estado natural se ha visto, por ejemplo, que hombres que fuman por más de 5 años les han tomado sangre y el nivel de pérdida del cromosoma Y aumenta. Y lo más interesante, dejan de fumar y se recuperan. Otro, hay 2 estudios muy interesantes en la India. 2 poblaciones en la India que viven en una región que naturalmente tiene mucha radiación del suelo, tienen mucho radón. Hicieron un estudio del cromosoma Y de los hombres que viven ahí en comparación de otros hombres que viven en otra parte de la India, y su cromosoma Y se encogió, y precisamente la región donde se encoge es el DYCY region, donde vienen estos link RNAs. Entonces, todas estas claves me dan a entender que son importantes en ciertos aspectos. Ahora, la aplicación, hasta donde yo sé, no. Okay. Había varios más. Sí.
En el contexto terapéutico, el delivery system, estas las moléculas, como perspectiva, ¿cómo cree que se pudiera implementar en el futuro? Intratumoral, de forma sistémica, porque en este caso las vacunas de COVID pues fueron como tal.
Sí, intramuscular. Sí, no, eso es algo muy importante que tenemos que ver. Ahorita en este momento, como modelo animal y tenemos el tumor técnicamente al lado, así en el flank del animal, pues va a ser fácil para nosotros inyectar las nanopartículas con el link RNA directo al tumor y hacerlo. Pero ya si estamos hablando ya a futuro, a una terapia, tenemos que ya empezar a ver qué tipo de nanopartículas se pueden, por ejemplo, utilizar como aerosol. Ahorita mucha gente está tratando de desarrollar nanopartículas que puedas tú distribuir por aerosol, y eso ya llegarían directamente al pulmón, y si tienes cáncer de pulmón, entonces llegarían directamente a esos lugares. Sí.
Mi pregunta es muy técnica. Cuando hacen la precipitación, ¿algún consejo para elegir los anticuerpos? Porque estaba viendo que hay unos como especiales para RIP y otros que son para IP. ¿Existe en su experiencia alguna diferencia o es igual?
No, es una muy buena pregunta. Sí, o sea, hay algunos anticuerpos que lo usas y son excelentes para Western blot o para RIP o para lo que sea, pero no funcionan para IP. Tienes que fijarte mucho en dónde se está uniendo el anticuerpo. Si es un RNA binding protein, tienes que checar que no es exactamente en la región donde está interactuando con el RNA. Entonces, si es la proteína como tal y es un anticuerpo que se pega a esa región, te puede dar unos Western blots muy bonitos, pero no va a funcionar para IP. Entonces, es algo fundamental que tienes que checar. Y aún así, checándolo así, tienes que hacer el experimento, porque muchas veces te dicen que funciona muy bien para IP, haces el experimento y no es cierto. Entonces, ¿alguien más? Voy a continuar porque ya más son unas pocas, y ya sé que son casi cuarto. De lo que les voy a platicar, es una pequeña historia de RNA circulares. Es un proyecto que interesantemente se los dejé a estudiantes de licenciatura, pensando que íbamos a sacar un paper muy chiquito, bonito, pero no con mucho impacto, pero finalmente lo que me han estado dando de datos me ha llamado mucho la atención y me ha gustado mucho. Okay, so I'm just going to jump to circular RNAs, yeah? Circular RNAs, people knew about them since 1970s and 1990s. People, by electron microscopy, knew that there was these circles of RNA, but they didn't know exactly where that was really coming from. Was not until 2013 in this paper, 2 papers back to back, that people by just serendipity identify circular RNAs. How? When they did an RNA-seq, and when they're looking at the reads of the RNA-seq, they identified that certain exons, that they should be before other exons, were after the exons. That didn't make any sense, like you can see here. The donors and acceptors were flipped. And turns out it's because the RNA-seq reads were here. And if you think of a linear RNA, this should be first, this should be second. But if this RNA is actually a circle, now they flip. So by this, they identify at that time close to 2,000 human circular RNAs in one paper. That was shocking. There was 2 back-to-back beautiful Nature papers. And now we know that we have close to 92,000 human circular RNAs. How many of them we know they have a function? I will be surprised if we know 30. No more than that. So how these RNAs, these circular RNAs come from what? Well, it turns out there are different ways they come. They can come from back splicing. This circularization come from the spliceosome. They can come from opposite orientation complementary sequences. They can come from competing for RNA pairing. They can come from intron bracketing binding. They can come from this quacking binding regulation. And they can even be regulated by ADAR proteins that they can change the nucleotides to form or don't form circular RNAs. But what do these circular RNAs do? In that paper that I just showed you, back-to-back papers, the second paper demonstrates that one of the circular RNAs are actually a microRNA sponge, meaning that they sponged microRNAs, and now the microRNA cannot bind to the thing they should be targeting. So that was the first evidence that these circular RNAs are functional. They identify now that these circular RNAs can bind not only microRNAs, they can bind RNA binding proteins. They can be actually assembly factories, so different RNA binding proteins, they need to bind to the circular RNA to actually have a specific processing. They can bind to other RNA binding proteins as allosterically. They can bind directly to messenger RNAs and regulate their expression. And more recently, there are evidence of these circular RNAs that can translate proteins. They can actually translate proteins using IRS regions. Now, my story is only going to be on HPV. And HPV, again, I'm not going to go into details. We know, of course, how important HPV is in human diseases. It's the most important infectious cause of cancer around the world, no doubt, even with the vaccine we have right now. Why is oncogenic? Again, we know that HPV produce these E6 and E7 oncogenes that normally in episomal state, they are regulated by the protein E2. But the thing that happens is in the moment the genome is integrated into the human genome, E2 is disrupted, and now E6 can target P53, E7 can target retinoblastoma, the 2 main tumor suppressor genes in our genome. Not only that, these E6 and E7 HPV proteins don't only target to P53 and RB, but they target several other proteins that are involved in immortalization, transformation, apoptosis, and so on. Yeah? So the big question is, if these circular RNAs are important in cervical cancer. So the first thing we did a screening of circular RNAs in cervical keratinocytes that we artificially introduced HPV16. And we, of course, identify a lot of circular RNAs were highly expressed after introducing the virus. Some of them were repressed. We focused in some of these guys like circ, IGF-2BP3. Remember I talked to you, IGF-2BP3, RNA binding protein? Well, it turns out the gene by itself produces a circle. And this is a circular RNA coming from that gene. There's other genes like circ-MEVIL2 that we saw overexpressed or repressed. We validate our data. We demonstrate that this circ IGF-2BP3 is increased in different cervical cancer cell lines that have HPV16 in an integrated genome or an episomal state compared to the normal cervical keratinocytes. Not only that, it's very hard to play with circular RNAs because, for example, if you want to knock down a circular RNA, you need to design an siRNA that binds to the end joining of the circle. You design any siRNA in any part of the circle, is not only going to target the circle, it's going to target the linear RNA that is coming from. So you need to target the end joining of the circle, and that region is when you have to design your siRNAs. So you don't have too much wiggle room. So we were actually able to design these siRNAs, able to show that we are able to knock down the expression of the circle without touching the expression of the proteins. Yeah? And by that, we also demonstrate that knocking down the circle, we saw changes in proliferation, and we saw changes, dramatic changes in migration. You can see here, Kasky cells, after we knocked down the circle, there was less migration. In SERPs 186, less migration. And that's it. This is all I have from circular RNAs. It is very, very, very preliminary data. My students right now are trying to use the other side of the coin. They're trying to overexpress the circles. That is also very challenging. So the way to overexpress circular RNAs artificially, we got a construct from a collaborator at Penn that he identified certain regions that they have to overlap each other. Anything inside of that regions, they can be circularized artificially. He gave us that cassette. We cloned it into a retroviral vector, and now we introduce this IGF to BP3 and overexpress it. So we have now this circular RNA overexpressed artificially, but my students right now are testing the different functions of the overexpression. And that's about it. Any questions, guys, for this? Yes. Adelante. Sí, sí.
Con los ARN circulares, entonces mi pregunta era si al momento de, ¿cómo decirlo?, de cortarlos o bueno, ya de desarmarlos, ¿tienen algún tipo de protección o ya se pueden mandar a degradar muy rápido?
Esa es una muy buena pregunta, lo que estaba platicando con Alfredo. Estos RNA circulares ahorita tienen mucha demanda, bueno, mucha gente en Estados Unidos están muy interesados en ellos, porque la mayoría de nuestros RNAsas adentro de nuestras células son exonucleasas, o sea que van a comerse el RNA por 5' o por 3'. RNA circulares no tienen endings. Entonces, si tú, por ejemplo, todos los RNAs lineales, su half-life, estás hablando en horas. Cualquier RNA dura 5, 6 horas, 4 horas, 8 horas. RNA circulares, se ha demostrado, y nosotros lo demostramos en el laboratorio, pueden durar 5 días. Son súper estables. De hecho, son más estables que proteínas. Y es por eso que ahorita mucha gente se está enfocando en RNA circulares como marcadores pronóstico y diagnóstico.
Gracias.
Acá.
Acá.
Mi pregunta es, de acuerdo a lo que entendí, y esto es completamente nuevo para mí, un mismo gene puede tener como resultado final este transcrito que se traduce a proteína o su forma circular. Y cada una de esas dos destinos finales, digamos, tiene una función diferente.
Exactamente.
Entonces mi pregunta es si se sabe el mecanismo para establecer la tasa, o sea, cuánto del transcrito va a irse por el destino 1 y cuánto por el destino 2, porque de eso dependerá la función del transcrito. Y cuando hacemos RNA-seq normal, estamos midiendo la combinación de ambos.
Claro, exactamente. Es muy buena pregunta. Es algo que tenemos muy pocos modelos. Y los pocos modelos que existen es cuando hay una autorregulación. Y eso es lo que nosotros estamos tratando de entender. Si tú ves a un transcrito en general, cualquier transcrito que tú te imagines, van a tener muchísimas regiones intrónicas que se va a perder. Entonces ya hay evidencia que en un solo transcrito puede producir a veces más de 10 diferentes RNA circulares, de uno solo. Entonces como tal va a producir una proteína, pero al mismo tiempo va a producir 5 o 10 diferentes RNA circulares. Al mismo tiempo, porque en el momento de splicing, en el momento que el splicing sucede, siempre pensamos que en el momento de splicing las regiones intrónicas que son esplaciadas, era basura. Pero muchas de esas, en vez de ser basura, se circularizan. Entonces es de donde la mayoría de los RNA circulares vienen, por regiones intrónicas que no son parte del RNA mensajero maduro. Entonces eso es lo que va a tener una función. Ahora, la tasa pues va a ser diferente dependiendo de la tasa de splicing, pero la forma de ver si esta tiene una función en comparación del otro, digo, los pocos ejemplos es cuando uno está regulando al otro, es un feedback loop. Y eso es lo que pensamos que está sucediendo con IGF-2BP3, porque IGF-2BP3, el RNA mensajero produce la proteína que es un RNA binding protein. Y no les dije, pero hay varias publicaciones donde dicen que IGF-2BP3, la proteína, lo han encontrado asociada con un montón de RNA circulares. Entonces, es obvio para mí que no nada más se está pegando otros RNA circulares, pero se está pegando su propio RNA circular. Y si se lo está pegando al mismo, entonces tiene que haber un feedback loop. Sí.
Del mundo de los RNA codificantes, ¿cree ustedes una jerarquía en cuanto a los distintos tipos que existen? Al haber estudiado, gracias. Al haber estudiado ya a los microRNAs, a los RNAs largos no codificantes, a los RNAs circulares, ¿considera que existe una jerarquía, que tenga un mayor peso un tipo que el resto?
No necesariamente. Unos, por ejemplo, cuando se descubren los microRNAs, mucha gente al principio no les dio mucha importancia, porque la mayoría de los microRNAs se les conoce como fine tunings. O sea que si te lo quitas o lo pones, no ves un efecto dramático. Pero lo que sí alcanzas a ver y se demostró es que como que regulan ciertas partes de la célula que tiene que tener una regulación muy fina. En los microRNAs ahí funcionan mejor. Ahora, sí hay ejemplos de microRNAs que si lo quitas, la célula se muere o le pasa algo. O sea, sí hay ejemplos. RNAs, long non-coding RNAs, son más funcionales. Tienen una función más específica. Abrir el DNA, interactuar con RNA binding proteins. O sea, ese tipo de interacciones son mucho más complejas que los microRNAs. Y los RNA circulares ahora estamos descubriendo que también son tan importantes o a lo mejor más importantes que los lineares. Entonces, hablar de jerarquías yo siento que es más hablar de diferentes tipos de pathways. Y ya no me dio tiempo, pero hay otro, el new kid on the block, el nuevo tipo de RNA que fue descubierto apenas hace 1 año y medio, que se llama glycoRNAs, RNAs glicosilados.
Okay.
Lo descubrió un cuate que se llama Ryan Flynn, era un postdoc a Stanford, ahora ya por lo que publicó, ya es profesor de la Universidad de Harvard, y estamos colaborando, porque él descubrió estos RNAs glicosilados, pero no tiene idea cuál es su función. Entonces nosotros estamos utilizando un modelo de head and neck cancer para identificar cuál es la función de ellos. Entonces, hablar de jerarquías yo creo que es un poquito artificial, es más que nada, cada uno tiene como que su propio nicho.
De acuerdo. Y un par de preguntas más, quizá la primera un tanto filosófica. Me gustaría escuchar su definición de conciencia. Para usted, ¿qué es conciencia, considerando todo lo que sabe actualmente de biología molecular?
Pues algo interesante es, para mí, conciencia no es más que una forma eso es algo muy personal, es una forma de energía que está dentro de las neuronas y que está regulada de cierta forma. Y la forma de prenderla o apagarla es como uno llega a tener cierto tipo de conocimiento. Pero eso es algo muy interesante y tampoco se los dije. La mayoría de los RNAs no codificantes, específicamente long non-coding RNAs, son de primates para arriba. Y lo más interesante, RNAs circulares. Si tú te preguntas cuál es el órgano que produce más RNAs circulares en todo nuestro organismo, cerebro.
Okay.
¿Por qué? No sabemos. Pero no me sorprendería que estos RNAs no codificantes sean parte de eso.
De acuerdo. Y la segunda pregunta es si considera que el cáncer humano es secundario a un desequilibrio en conciencia.
Sí, yo creo que sería un poquito más filosófico y no quisiera yo responder a eso.
De acuerdo. Muchas gracias.
No hay problema.
Sí, muchas gracias por la presentación. Me llamaba la atención en los resultados con los largos no codificantes, que partiendo del análisis de las líneas resistentes y no resistentes, la ausencia del cromosoma Y era definitivo, era un resultado de cero, sí o no. Y cuando lo regresas a las células, el fenotipo no cambia de esa forma, ¿sí? Es decir, no se desaparece o aparece la función.
Claro.
Es decir, queda como a la mitad. ¿Esto qué quiere decir? ¿Significa que es cosa de dosis o que hay otros componentes adentro del cromosoma que responden a esto? Y la otra cosa que quería preguntar era, ¿la dosis de radiación que se utiliza con estas células es de algún modo parecido a lo que usamos con los tumores reales?
Sí, sí, de eso, en ese aspecto fuimos muy cuidadosos de tratar de ver en las líneas celulares que tuvieran las mismas dosis de radiación que lo que normalmente los clínicos dan para los pacientes. De eso fuimos muy cuidadosos desde el principio. Entonces siempre le hemos dado el número de grays a estas células para que más o menos se asemejen a lo que se da un tratamiento clínico. Ahora, en cuanto a lo de la recuperación de radiosensitividad, tiene mucha razón. Pueden ser las dos cosas. Puede ser de que sea algo de dosis, y eso tendríamos que verlo, por ejemplo, con diferentes tipos de promotores de diferentes construcciones que podemos modificar o alterar y ver si tenemos un plateau y ya más, aunque le metamos más expresión, ya no suba más. Entonces nos diría que llegó a su límite. Pero para mí yo siento que hay otros RNAs no codificantes en el cromosoma Y que potencialmente también pueden estar funcionando como estos. Y como solamente introdujimos los RNAs, los long non-coding RNAs que estamos enfocados ahorita y no los otros, a lo mejor por eso no recuperamos completamente el fenotipo.
Gracias.
¿Nadie más?
Hola, buenos días. Por acá. Primero que nada, felicidades por el trabajo.
Gracias.
Estupendo trabajo, ¿verdad? La otra parte sobre los RNA circulares, que a final de cuentas con esta parte de que se estabilizan y que pueden entonces ser traducidos por el ribosoma. Entonces, ¿la estabilidad de la proteína que se produce es la misma si el RNA está circularizado o está linealizado?
Sí, eso es muy buena pregunta, porque gente en biofarmacéutica está muy interesado en esto. Y la estabilidad de la proteína es la misma, si viene de un círculo o si viene de un RNA lineal. Lo que sí es que el círculo, al estar circularizado, tiene mayor capacidad de producir esta proteína que uno lineal. Y es por eso que esa estabilidad del RNA circular ha tenido muchísimo, ahorita muchísimas compañías están tratando de usarlo como modelo.
A final de cuentas tiene un proceso de transcripción y luego a la traducción. No, pues es el RNA directamente. No, pues ya te brincaste. Maravilloso.
Sí.
Sí, la tasa de transcripción, bueno, la traducción que tendrías te va a dar un rendimiento mayor.
Mayor.
Precisamente por eso.
Así es.
Muy
así
bonito.
es. Sí, de hecho, tengo un compañero que trabaja en colaboración con Pfizer y con Moderna, y ahorita están ellos precisamente tratando de desarrollar vacunas con RNA circulares.
Sí, porque incluso podrías tener polirribosomas que están sobre el mismo RNA, y aunque no tengas tantas copias del RNA circularizado, pues puedes estar traduciendo.
Exactamente.
Más que la tasa de traducción, es la estabilidad del mensaje. O sea, vas a tener una molécula más estable en el tiempo
que va a permitir que no le tengas que estar metiendo el transcrito lineal más repetitivamente. El transcrito lineal se te va a degradar en varias horas, mientras que el transcrito circular puede quedar ahí 3 días siguiendo produciendo el biológico, ¿no? Precisamente porque tiene una estabilidad mayor. Entonces la tasa de traducción potencialmente es la
muchísimo
misma,
mayor.
Pero no determinado porque sea distinto a nivel ribosomal, sino simplemente por el tiempo que dura traduciéndose ese circular en relación al lineal.
Exactamente.
Okay.
Y podría hacerse en otras células que no fueran células humanas, eucariontes.
Por supuesto. Sí. Bueno, no sé si en procariontes se podría hacer eso, pero en cualquier célula eucarionte sí, mientras tenga un IRS y un ribosoma que lo reconozca.
Gracias.
Yo, o sea, en realidad, potencialmente cualquier transcrito, cualquier gen podría generar circulares. Sin embargo, si tú ves los números, pues sería en varios órdenes de magnitud superior el número de circulares que de transcritos lineales. Pero no es el caso.
No.
Entonces, ¿qué estará definiendo realmente quién sí lo hace y cuándo lo hace?
Es algo que no conocemos realmente. Hay genes que nada más producen un RNA circular, hay genes que no producen ningún RNA circular y otros, como les dije, que producen 10.
Varios, exacto.
Lo que sí sabemos es que cuando uno está trabajando con ellos también tiene que tener mucho cuidado cuando uno dice, este es un RNA circular. ¿Por qué? Porque en el momento de splicing, y si ustedes recuerdan, se corta primero de una parte del intrón y esa parte del intrón se pega a la otra parte que va a ser cortada y forman un lariat. O sea, es como casi un círculo, pero tiene una colita. Y estos lariats, tú puedes usar primers que tú crees que es un RNA circular y lo puede amplificar y no es una RNA circular, es un lariat. Entonces tienes que hacer diferentes tipos de experimentos para demostrar que es un RNA circular real y no un lariat. Como por ejemplo, actinomycin D, utiliza actinomycin D para parar transcripción y los lariats tienen un promedio de vida de 8 a 12 horas. Si es algo que te dura 2 o 3 días, lo más seguro es que sea un RNA circular. Pero sí.
Yo otra vez.
Adelante.
Es que fíjate que sí me ha llamado muchísimo la atención este tema de que aumenta el número de cromosomas Y en las células que son quimioresistentes, ¿no? Entonces estaba pensando si había
Son
neuploidías.
quimiosensibles.
En las quimiosensibles, sí. Si había neuploidías o cuando le insertan a el largo no codificante a las células, también las vuelve sensibles. Entonces estaba tratando de pensar qué mecanismos, nos mostraste una lista de genes que están alterados en su expresión, etcétera, pero qué mecanismos podrían estar implicados. Y pensaba si la neuploidía o tal vez a la ganancia de mutaciones o alteraciones en las mitocondrias o algo que las vuelve ahora radiosensibles a estas células, ¿no? Esa es como algo que me ha llamado mucho la atención. Y la otra es que también respecto a los circulares, nos mostraste un ejemplo de 2 genes que se ven siempre afectados en HPV, ¿verdad? Y bueno, pues la inserción del virus, etcétera. Y comentábamos con el doctor si hay alguna relación entre en dónde se localizan estos circulares y regiones que hayan sido o que deriven de virus, inserciones, secuencias dentro del genoma que correlacionen y que podrían de alguna manera tener algún origen viral estos.
Sí, no, son 2 muy buenas preguntas. Déjame contestar la primera en cuanto a los link RNAs. No tampoco les mostré estos datos, pero los tenemos. El simple hecho de meter los link RNAs artificialmente, algo que descubrimos es que H2AX, que es un marcador de corte de doble cadena de DNA, es un marcador que te dice que el DNA se está cortando. En el momento que nada más lo introducimos sin necesidad de radiación, vemos que este H2AX se incrementa. Y lo que a mí me da a entender significa es que simplemente parte de la radiosensitividad que da estos link RNAs es a lo mejor dar sensitividad al DNA a que se corte más fácil. ¿Cómo es el mecanismo? No sabemos todavía. Ahora, la otra del circular RNA. Sí, sería muy interesante ver dónde se está integrando el virus en el genoma humano y ver si está cerca o lejos donde se están produciendo estos RNA circulares. Pero no sé si se fijaste y no sé si lo dije muy rápido. Una de las líneas celulares que checamos, que es la W122863, esta es una de las pocas líneas celulares de HPV que todavía el virus es episomal, no está integrado en el genoma. Y aún así vemos el incremento de circ-IGF-2BP3. Entonces, para nosotros fue importante ver eso, que no es la inserción como tal, sino simplemente la presencia del virus. Bueno, pues muchísimas gracias.
Muchas gracias, doctor Martínez. Les pedimos que permanezcan un momento, por favor, para darles unos...